morning and welcome to the University of Cape Coast College of Distance Education program Bio 208 Population Genetics and Evolution Unit 1 Session 1. What is population genetics? Your course instructor is Dr. John Eko Imbir Amwa of the Faculty of Science Education, Department of Biology Education, Winneba. Population genetics is concerned with the origin, amount, and distribution of genetic variation present in populations of organisms and the fate of this variation through space and time. Variation in genes through space and time constitutes the fundamental basis of evolutionary change, and indeed its most basic sense. Evolution is the genetic transformation of reproducing populations over space and time. Then again, population genetics is therefore at the very heart of evolutionary biology and can be thought of as a science of the mechanisms responsible for microevolution, evolution within species. And many of these mechanisms have a great impact on the origin of new species and on evolution above the species level. So most humans are susceptible to HIV infection. However, some people seem to be able to avoid infection despite repeated exposure. Some resistance is due to a rare allele of a gene that codes for the CC chemokine receptor type 5 protein. The CC chemokine receptor type 5 is a cell surface protein that is a co-receptor for the HIV virus. Then it binds to the cell membrane of macrophages and T cells. Some people have a form of the gene encoding CCR5 that includes a 32BP deletion that results in a dysfunctional protein. This allele is called chemokine receptor type 5, 32BP deletion. So individuals who inherit two copies of the deleted allele have no CCR5 co-receptors on the cell membrane of quiet blood cells. These homozygotes are highly resistant to HIV infection. Will the global HIV epidemic result in increasing frequencies of the CCR5 beta 32 allele? If so, how soon will this happen? Another Northern European populations carry a higher frequency of this allele than do Asian or African populations. So is this difference due to chance or to the fact that CCR5 32 also increased resistance to some previous epidemic that occurred in Northern Europe, that's a plague, smallpox, etc. So these kinds of questions that population genetics is designed to answer. This discipline describes the behavior of alleles in populations by focusing on the forces that can cause allele frequencies to change over time. Allele frequency change over time is simply a definition of evolution. So population genetics is that branch of genetics that is concerned with the evolutionary processes of natural selection, genetic drift, mutation, migration, and non-random mating. Population genetic approaches can be used to understand the consequences of these processes individually or in combination. Some basic terminologies in population genetics. Allele, one of a pair or series of alternative forms of a gene that occurs at a given locus in a chromosome. Allele frequency, this is the proportion of one allele relative to all alleles at a locus in a population. It is also called gene frequency. Assortative mating, non-random mating on the basis of phenotype. In positive associative mating, the partners are chosen because they are phenotypically similar. Balancing selection, a general term for selection that results in the maintenance of different alleles. This could result from heterozygote advantage over dominance or frequency-dependent selection. Biogeography, the study of the biogeographical distributions of organisms. Biogeography involves a range of scientific disciplines that includes geography, geology, and biology. Bottleneck, a severe temporary reduction in effective population size, sometimes leading to genetic drift cladistics, a classification system that classifies organisms on the basis of historical sequences by which they have diverged from common ancestors. Co-dominant alleles that produce independent effects when heterozygous. Co-evolution, the coordinated evolutionary change in two or more species in response to the presence of other species. Diploid, an organism or cell with two sets of chromosomes or two genomes. Directional selection, selection for a higher or lower value of a character than its current mean. Disassociative mating, non-random mating on the basis of phenotype. In disassociative or negative associative mating, the partners are chosen because they are phenotypically dissimilar. Disruptive selection, selection in favor of two or more modal phenotypes and against those intermediate between them. It is also called diversifying selection. Dominant, a condition in which one member of an allele pair is manifested as it determines the phenotype to the exclusion of the other. Elicitor, a molecule that binds to a receptor and initiates the plant defense response system. Elicitors are often encoded by a virulence allele of the pathogen in a gene or for gene interaction. Epidemiology, the study of the development of disease in populations. Equilibrium, a state at which there is no change in the genetic constitution of a population. An equilibrium may be stable, unstable, or neutral. Evolution, the change over time of the population of the proportions of individual organisms differing genetically in one or more traits. These changes are brought about by the origin and subsequent alteration of the frequencies of alleles or genotypes from generation to generation within populations. The fitness, the average contribution of one allele or genotype to the next generation or to the succeeding generations compared with that of other alleles or genotypes. Fitness costs, the reduction in fitness compared to the population mean fitness due to the presence of a particular allele or phenotype. Founder effect, the principle that the founders of a new colony carry only a fraction of the total genetic variation of the source population. Gametic equilibrium, the random association of alleles at different loci into gametes. At equilibrium, the frequency of the gametes is equal to the product of the frequencies of the constituent alleles. Gene diversity, a measure of the total number and frequency of alleles within a population. Gene flow, the incorporation of genes into the gene pool of the one population from one or more other population. It can be used synonymously with migration. Gene for gene, a model to explain the genetic basis for the interaction between plants and parasites introduced by H.H. Floor. For each gene determining resistance in the host, there is a corresponding gene in the parasite with which it specifically interacts. Genetic bottleneck, a temporary reduction in population size from which future generations are derived. Genetic drift, changes in allele frequency in small breeding populations due to chance fluctuations. Genotype, the set of genes possessed by an individual organism. Haploid, an organism or cell having only one complete set of chromosomes or one genome. Effective population size, the size of breeding individuals in a population that contribute offspring to the next generation. Population, a population is a group of organisms of the same species living within a prescribed geographical area. The area is usually determined to be of a size within which individuals are likely to find mates. Geographically widespread species are often subdivided into more or less distinct breeding groups that live within limited geographical areas. And these groups are called subpopulations. The complete set of genetic information contained within the individual is called the gene pool. The gene pool includes all genetic loci and all the alleles for each locus present in the population. The gene pool of a population can be described and characterized by certain statistical properties or geographical areas. The area is usually determined to be of a size within which individuals are likely to find mates. Geographically widespread species are often subdivided into more or less distinct breeding groups that live within limited geographical areas. These groups are called subpopulations. The complete set of genetic information contained within the individual population is called a gene pool. Gene pool includes all genetic loci and all the alleles for each locus present in the population. So the gene pool of a population can be described and characterized by certain statistical properties. Determination of phenotype by two alleles at a single locus. We'll begin our discussion of population genetics by considering an analysis of the local population with respect to a phenotype determined by two alleles at a single locus. So the human MN blood group is an extremely simple system. It is characterized by a single locus with only two alleles, M and N, sometimes referred to as LM or LN. The alleles are co-dominant. Both alleles are detectable in heterozygotes as seen in tables 1 and 2. Both heterozygosity and polymorphism are used as measures of genetic variation within populations. So different alleles within populations can be identified by phenotypic effects as through color, size, shape variance disorders like color blindness or Huntington's disease, by variation in protein structure, standard blood type assays, protein electrophoresis, or by variation in DNA sequence, restriction site variation, variation in number of repeats in repetitive DNA segments, DNA sequence variation. So the blood types we have, the phenotypes or the genotype, we have M, MM, LM, LN. We have MN, we have another MN as a genotype, and in the blood type we have N and double NN. So we have 298 M, we have 298 MM. The alleles are 596, and N alleles there is none. The next we have 489 MN, 489 MN, 489 alleles, 489 N alleles. So in the next we have 213 N, 213 MN, and the alleles for that is 0. We have 426. The total we have 1000, 1000, 1085, 915. Table 2. So the genotype frequency is the relative proportion of the genotype. Proportion of MN genotype equals 298 over 1000, giving us 0.298. Proportion of MN genotype 498 over 1000, giving us 0.489. Proportion of NN type, you have 213 over 1000, giving us 0.213. Total is 1. The allele frequency of the M allele is the relative proportion of the allele. Proportion of M allele equals 1085 over 2000, giving us 0.5425. Proportion of N allele is 915 over 2000, giving us 0.4275. Total is 1.000. If we let P equals the allele of frequency of M and Q the allele of frequency of N, then Q equals 1 minus P. So population genetics defines the origin, amount, and distribution of genetic variation present in populations of organisms, and the fate of this variation through space and time. The variation in genes through space and time constitute the fundamental basis of evolutionary change. Indeed, in its most basic sense, evolution is a genetic transformation of reproducing populations over space and time. Population genetics is therefore at the very heart of evolutionary biology and can be thought of as the science of the mechanisms responsible for microevolution, evolution within species. The complete set of genetic information contained within the individual in a population is called the gene pool, and it includes all genetic loci and all the alleles for each locus present in the population. Different alleles with populations can be identified by phenotypic effects, by variation in protein structure, or by variation in DNA sequence.